Assalamu alaikum. Today we are going to start chapter number 15, which is control and coordination. And the learning objectives for today are compare the nervous and endocrine system as communication systems and coordinate responses to changes in the internal and external environment. Describe the structure of a sensory neuron and a motor neuron. Outline the roles of sensory receptor cells in detecting stimuli and stimulating the transmission of nerve impulses in sensory neurons. And the example of chemoreceptors from the book will be given. Describe the functions of sensory relay in motor neurons in a reflex arc. Describe and explain the transmission of an action potential in a myelinated neuron and its initiation from a resting potential. The importance of sodium and potassium ions in impulse generation should be emphasized. Explain the importance of myelin sheath, saltatory conduction in determining the speed of nerve impulses, and the refractory period in determining the frequency. Describe the structure of cholinergic synapse and explain how it functions, including the role of calcium ions. Outline the role of synapses in the nervous system in allowing transmission in one direction and in allowing connections between one neuron and many others. However, summation, facilitation, and inhibitory synapses are not required. So these are all learning objectives as per our CAI specifications. All right then, so let's start with nervous coordination. So we are going to first discuss what is actually a nerve impulse. So by definition, a nerve impulse is an electrical signal that travels along a nerve fiber in response to a stimulus and serves to transmit a record of sensation from a receptor or an instruction to act to an effector. So these are the medical definitions of nerve impulse. And the second definition is the propagation of an action potential along the length of a neuron. So yeah, so as we said, a nerve impulse is an electrical signal that travels along the nerve, right? But it is not quite electricity, which is actually the flow of electrons, right? So in a nerve impulse, the word potential is actually used, right? The word that we use is potential. And this is used to describe the movement of charged particles across the membrane of the cell. So these charged particles actually flow as a wave, right? And they flow down the axon, moving in and out of the cell. So here, if you're looking at the positive ions, they're going in the cell, right? And the negative ions are going out. So it is actually the movement of charged particles in and out, right? And mostly it is all the positive charges, right? They mostly move in, right? The negative charges, they don't go out as much as compared to the positive ones. So that's why it is known as a potential. So here also you see that throughout along the length of the axon or neuron, charges go in and they go out, right? All right, so now let's talk about the nervous system. So the human nervous system consists of two parts, central nervous system and peripheral nervous system. So central nervous system, as the name itself implies, consists of brain and spinal cord, while the peripheral nervous system consists of all the nerves in the body, which are actually the cranial nerves as well as the spinal nerves. So have a look here. This is the central nervous system that includes the brain and the spinal cord, right? But all the peripheral nerves that you see, this makes the peripheral nervous system. So these are cranial nerves, right? And these are the spinal nerves. So these two constitute the human nervous system. So a nervous system allows us to make sense of our surroundings and then respond to them and to coordinate and regulate body functions. So this information is actually sent through the nervous system as nerve impulses, as we were discussing a while back, electrical signals that pass along the nerve cells as neurons. Now, a bundle of neurons is known as a nerve, and these neurons coordinate the activities of sensory receptors, for example, those in the eye, then the decision-making centers which are in the central nervous system, right? And then the effectors, which can be muscles or glands. So this is how the nervous system works. So in the previous chapter, we have already discussed about endocrine system, we discussed about the hormones. So let's just talk about a few differences between a nervous system and the endocrine system. So the nervous system actually involves the brain, spinal cord, nerves, and neurons, while the endocrine system involves the glands, right? When we talk about the type of message, so a nervous system carries an electrical impulse, right? But in terms of endocrine gland, it's a chemical hormone, right? So this electrical impulse in a nervous system is carried by the nerves or neurons, while in endocrine system, this chemical hormone is sent through the bloodstream. Now, these nerves and neurons in a nervous system, they act on the muscles or the glands, right? While these hormones through the bloodstream, they act on the target cells in specific tissues. Now, since these nervous system actually involves electrical impulses, they are very fast. While endocrine system, the actually hormones take time to travel through the blood to reach the target cells. So this is comparatively slower. If you talk about the length of the effect, so nervous system, the effect is short until electrical impulses stop. But in an endocrine system, the effect is rather long until the hormone is broken down eventually. Okay, there's a lot. Let's talk about the neurons now. So neurons have a long fiber known as axon, which is insulated by a fatty sheet with the small uninsulated sections along its length called as nodes of Renvier, right? So we'll just have a look at the picture. See, this whole is a neuron. And here what you see is a cell body. And this part here that you're seeing is the axon, right? And it is covered with this myelin sheath, the one that is in yellow. So it is insulated by myelin sheath. And then these are here disconnected at the nodes of Renvier. The part where there is no myelin sheath, this part here, this part here, this part here. So these are known as nodes of Renvier. So this myelin sheath is made of specialized cells, which are known as Schwann cells, right? So this myelin is made when Schwann cells wrap themselves around an axon along its length. So have a look here. This is an axon, right? And this is the myelin sheath. This is a Schwann cell which wraps around the axon, right? And this forms a myelin sheath along the axon, right? That actually then serves as insulation. So because of this myelin sheath, electrical impulse does not travel like as it is, right? So it actually jumps. So it jumps along the nodes of Renvier, right? The myelin sheath actually wraps around a part of axon, right? Then this is a part which doesn't contain any myelin sheath. Then after some distance, we again have a myelin sheath. So this is how the electrical impulses jump actually. And this actually helps in saving time. As the impulses jump around one node to another, so this actually helps in saving time. And the impulse is passed on quickly from one node to the next. Now, if you look at the cell body here, it contains many dendrites. So these branches that you're looking at, these are cytoplasmic extensions. And these actually connect this cell body to other neurons, right? And increase the surface area as well. So these actually make communication easy. And it becomes easy to receive the nerve impulses. So this is how these neurons are connected. So this is a cell body. You're looking at a nucleus. These are the mitochondria, right? Because we need ATP as well here. And there is a lot of endoplasmic reticulum here as well for protein synthesis. And here, if you look at this part here, so these are again dendrites, and they form connections with many other neurons. So they would actually connect with the next neuron there, right? So this is known as a nerve ending as well. So we have three main types of neurons: sensory neuron, relay neuron, and the motor neuron. So there are structural differences between these. This is a sensory neuron. This is a relay neuron. And this one here is actually the motor neuron. So the function of sensory neurons is actually to carry impulses from receptors to the central nervous system. So receptors can be eye, it can be skin, so anything that can sense a stimuli, right? So it sends those impulses from the receptors to the central nervous system, which can be based on the spinal cord. Now these relay neurons, actually, these are also known as intermediate neurons, right? These are found entirely within the central nervous system, right? And when we say central nervous system, so it might be the brain or the spinal cord. And what they do is they connect sensory and motor neurons. This is what they do. That's why they're known as intermediate neurons. They connect sensory and the motor neuron, right? And then when it comes to motor neuron, it carries impulses from the central nervous system to the effectors. And the effectors can be muscles or glands. See, so there are three types: sensory, relay, and motor. So as we discussed, there is actually some basic structural difference. So a sensory neuron has the same basic structure as a motor neuron. So this is a sensory neuron, right? But it has one long axon, right, with a cell body that may be near the source of the stimuli, or is actually in a swelling of a spinal cord, which is known as ganglion, right? So this is a ganglion, right? So here you can see this is known as the dorsal root ganglion, and this is known as the ventral root ganglion. So ganglions are usually group of neuron cell bodies, right? So if you're talking about a sensory neuron, so these sensory neuron cell bodies are actually found here in the ganglions, right? So these are clusters of nerve bodies which are positioned along the spinal cord at the dorsal root. So this is known as the dorsal root and the ventral root of the spinal nerve, right? And these cell bodies actually are of the sensory nerve fibers that carry impulses from the stimulus or from the receptors towards the central nervous system, right? So here, if you see again, right? So these are dendrites, axon, cell body here somewhere in the middle, right? And then axon extends, and these are the dendrites now. Now, this is actually the relay neuron or the intermediate neuron, which is found entirely in the central nervous system, which can be brain and the spinal cord. So the impulses would be brought through the sensory neurons, right? And this would connect to the relay neuron. So relay neuron, you see these are a lot of branches throughout. Again, they're extending from the cell body. So this is where these connections are made. So it actually acts as a communication between the sensory neuron and the motor neuron. So if you just imagine there's a sensory neuron here, and it is connected to this relay neuron. So this relay neuron would then be connected to the motor neuron. Okay, then so let's talk about the structure of a motor neuron now. So motor neuron transmits impulses from the brain or the spinal cord to a muscle or a gland, right? The cell body of this neuron lies within the spinal cord of the brain, and the nucleus of the neuron is always in the cell body. So if you look here, so this is how a motor neuron looks like. So this is the cell body, and here you can see there's a nucleus here, and here you can see these are the cytoplasmic projections. Unlike the sensory neuron, if you remember the sensory neuron that we just saw, right, the cell body was there, but there was no dendrites attached to it, right? And it was there somewhere along here in the axon, along the axon, right? But a motor neuron actually, it has a large cell body with a nucleus and the cytoplasmic branches known as dendrites. And often dark specks of endoplasmic reticulum are seen here actually for synthesizing protein along with a lot of mitochondria, right? So here these cell organelles can be found, endoplasmic reticulum as well as the mitochondria. Then you can see this is the axon, right? This is the myelin sheath, right? The nucleus of the Schwann cell. Then these are the nodes of Ranvier, as we just discussed earlier. And this is how the nerve impulse will jump from one node to the next, right? And it will be a quick response. And now this axon can, maybe it can connect to another neuron, or it can connect to the end plate. For example, it may connect to the muscle or the gland, as it will carry the instructions from the brain, right, towards the muscle or the gland, right? So these are the terminal branches, and this is the synaptic knob. Through here, actually, these neurotransmitters actually will be released. So we'll talk about that in a while. But for now, understand this is how a motor neuron looks like. Now, another difference between a motor neuron and a sensory neuron, like besides the cell body not having dendrites in a sensory neuron, the axon of a motor neuron is also much longer, and it conducts impulses over long distances. So a motor neuron whose cell body might be in the spinal cord, axon can be running all the way to one of your toes. So they're extremely, extremely long, right? And within the cytoplasm, we just actually saw the picture here also, you'll find a lot of mitochondria here as well, because they produce ATP, right? And for these action potentials to happen, right, we need this ATP here. So just have a look at the axon of a motor neuron, see how long it is, and compare it with this axon of the sensory neuron, right? So these mitochondria are found along the cell body as well, And a lot of mitochondria are found here at the terminal branches as well. Right? And together, these mitochondria, along with the vesicles, which contain a neurotransmitter, right? These vesicles, they're involved in passing impulses to the effector cell, which can be muscle or the gland, right? So we have a lot of mitochondria here because energy is required, ATP, right? For these nerve impulses to travel here, right? And then we have a lot of mitochondria here as well, because through this, these neurotransmitters are actually released into the synaptic cleft. So we'll talk about that in a while. So this is the myelin sheath, the one that you see here. So for most of the length, these axons are actually covered by these myelin sheaths. So axons of motor neurons, they're protected within the nerves. So if you look at a cross section of a nerve, which is full of neurons, so you will actually see that these are surrounded by thick dark rings. Now we'll have a look at the photomicrograph also in a while. So these dark circles are known as myelin, which are made by Schwann cells, which are specialized cells. And just have a look here. So see, this is a photomicrograph. And if you're looking at these circles, which are surrounded by these darker rings, these are the axons which are surrounded by the myelin sheath, right? So not all are covered, around 1/3 are actually covered or are known as myelinated, right? So these darker circles that you're seeing are axons which are covered by these, right? And this red part that you're looking at here is a perineurium, right? So several groups like these, like this is a perineurium. So you'll see a lot of axons here. So there will be another group like that. So a number of these groups actually make a nerve. So have a look at this picture. So these are axons. This is a perineurium, right? And then you can see one here, one here. So a lot of these groups actually make one nerve, right? So this is one nerve, and it consists of these groups of axons here that you see, right? So this is actually what a nerve fiber looks like. So about 2/3 of our motor and sensory neurons are unmyelinated and 1/3 are myelinated. So these Schwann cells, they spiral around these axons, right? So they spiral around, if you look here. So you look at the arrow as well. So they make many layers of its cell surface membrane, and the sheath that they form is what we call as myelin sheath, right? So this myelin sheath is actually made of lipids and proteins. So yes, this is a cell surface membrane. So you can imagine why it consists mostly of lipid together with some proteins, right? So this sheath actually affects the speed of conduction of a nerve impulse, right? So, and these actually, the uncovered areas of the axon, which we call as node of Ranvier, they actually occur every 1 to 3 millimeter in human neurons, but these nodes themselves are very, very small. They're around 2 to 3 micrometers long, but they occur every 1 to 3 millimeter. If you look at this picture again, these nodes would occur every 1 to 3 millimeter, but if you talk about their size, so they're around 2 to 3 micrometer, right? But they would occur every 1 to 3 millimeter. Okay, now so let's talk about the reflex arc now. So these sensory neurons, which are connected through these relay intermediate neurons to the motor neurons, like so all these three, they work together to bring about a response to a stimulus, right? So what is a reflex arc? A reflex arc is a pathway along which impulses are transmitted from a receptor to an effector without involving the conscious regions of the brain, right? So signals, they would go to the spinal cord, which actually acts as a relay. They go to the brain, but since it takes time for the signal to go to the brain as well, the spinal cord is nearer, right? So the signals will be relayed. So that is what we call as a reflex arc. Actually the response, we get the response without involving the conscious regions of the brain, right? So that's the reason the reflex response is very, very quicker than any other type of nervous response. So the examples of simple reflex actions are removing the hand rapidly from a sharp or hot object, blinking, focusing of the eye on an object, controlling how much light enters the eye. So these are all reflex mechanisms of our body. So they do not require any conscious efforts on our part. So this is how it works. See, this is the spinal cord. And as this impulse goes to the spinal cord, it is relayed through the spinal cord. See, the spinal cord actually coordinates, okay, and then sends instructions to the muscles to act, right? It would go to the brain. Remember, it would go to the brain, but it takes time, okay? But since this is dangerous, right? So spinal cord would actually quickly act as a relay, right? And send the instructions to the effectors. So what is happening in this case is, this is a pin, which is a stimulus. It is detected by pain receptor in the skin. So let's say this person actually has, right, stepped on a pin. So there's a pain receptor that has been activated. So it sends the signal through the sensory neuron, right? And it sends it to the spinal cord here. So the spinal cord actually acts as a coordinator. Now these electrical impulses are passed on to the relay neuron, which is here in the spinal cord, right? So the one in orange that you see, this is the relay neuron, right? The relay neuron then connects to the motor neuron, the one that you see in green here. So it connects to the motor neuron and it passes the impulses, and the motor neuron then carries these impulses to the muscle in the leg, right? Which is the effector. And what these effectors do, they actually cause this muscle to contract, right? So definitely the person will pull the leg up and away from the sharp object. So that would be the response, right? So as we were discussing, the impulse is also sent to the brain, but the brain is not involved in coordinating the response. The response is being coordinated by the spinal cord here, right? So this is what is a reflex arc, right? So this is the flow chart. This is how it starts. There's a stimulus, right? So it is sensed by the receptor. The receptor, okay, takes it to the sensory neuron. Okay, then this passes to the relay neuron, the motor neuron, effector, and finally we get the response. So this is what we call as a reflex pathway. Okay, so let's talk about the sensory receptor cells. So any cell that responds to a stimulus is what we call as a receptor cell, right? It responds to a stimulus. And these receptor cells are actually transducers. What they do, they convert the energy in one form, right, to another form. So this is what a transducer does. So these receptor cells, they act as transducer. They convert energy in one form, which can be light, heat, or sound, into an energy in an electrical impulse. That's why we call them transducers. They convert this energy, which can be light energy, heat energy, or sound. These are all energies. And it converts that into electrical impulse, right? And this electrical impulse is then taken through this sensory neuron, right? So these receptor cells are often found in the sense organs. So the sense organs like eye. So we have light receptors here in the eye, right? So these light receptors in the eye, and then chemoreceptors in the taste buds. They're specialized cells. So these are specialized type of cells that detect a specific type of stimulus and influence the electrical activity of the sensory neurons, right? So these are specialized cells. And other receptors, such as some kinds of touch receptors, they're just the ends of sensory neurons themselves. So these touch receptors, they are the ends, right? But when we talk about the light receptors or when we talk about the chemoreceptors and the taste buds, they're specialized cells. So within these receptor cells, when they're stimulated, they become depolarized, right? So their resting potential actually changes. We'll talk about that just in a while as we discuss action potential. So what happens, these receptor cells, as they're stimulated, they become depolarized. So if the stimulus is very, very weak, the cells are not sufficiently depolarized and the sensory neuron is not activated to send impulses. So actually a threshold has to be reached. So if the threshold is not met, no activation of these nerve fibers will not take place. So no nerve impulse will be sent. But if the stimulus is strong enough, it crosses the threshold, only then the sensory neuron will be activated and it will transmit those nerve impulses to the central nervous system, right? So this is the part where actually an action potential will be generated. So we'll talk about that in detail in a little while, but just understand an impulse will only be generated and that sensory neuron will only be activated, right, if the threshold is met. If that certain threshold is not met, no sensory neuron will be activated and no nerve impulse will be sent to the central nervous system. The stimulus has to be strong enough. So here in this picture, we are looking at two types of receptors. So these are the specialized type of receptors that they're showing you here. So this is the stimulus, this is a cell body, right? And this is actually what we call as a presynaptic membrane, this part here. And this is what we call as a postsynaptic membrane. So this is the part of the sensory neuron, okay? So postsynaptic membrane. And it is actually through these vesicles, the neurotransmitter will actually be released, right? So this is a specialized type of receptor. And the others can be simply the ends of a sensory neuron, as we just discussed about the touch receptors there. Okay, so these are two different types of receptors. So this is a simple end of a sensory neuron. This one is a specialized cell. Okay, so let's discuss an example. So this is a sequence of events that results in an action potential when it occurs in a sensory neuron. So the surface of our tongue is covered with many small bumps, which are known as papilla. So this is the tongue, right? And this is a close section here, so in large view. So these bumps are what we call as papilla. So these papilla, they contain many taste buds. These papilla, they contain many taste buds. So each taste bud contains many receptor cells, which we call as chemoreceptors, right? So we call them chemoreceptors because they're sensitive to chemicals in the food and drinks. And each chemoreceptor is then covered with receptor protein. And we have different receptor proteins for detecting different chemicals, right? So these chemoreceptors in the taste buds, they can detect salt, right, which is sodium chloride, and then they would respond directly to the sodium ions. So we have different type of receptors. We have receptors for sweet, for bitter, for salty things, right? So a lot of receptors are there. So right now we're talking about the receptors that can detect salt, sodium chloride. So if salt is present in the food, which becomes dissolved in the saliva after being eaten, or any liquid that has been drunk, so what happens, these sodium ions, they diffuse through highly selective channel proteins in the cell surface membrane of the microvilli of the chemoreceptors. So remember we discussed that these are specialized type of cells, so they have specialized features. So these chemoreceptor cells, they have microvilli. Microvilli increase absorption. So what happens because of the sodium ion diffusion, this leads to depolarization of the chemoreceptor cell membrane. Now, when we talk about depolarization, we're talking about this, right? These charges will actually change places. So see, this will be a flow of charges here. So positive ions will move in, negative charges will move out, right? So this is actually what is causing depolarization, right? This is what is generating an action potential. So what happens because of this, now we have increase in the positive charge inside the cell, right? And now this is what is known as the receptor potential now. So if there is sufficient stimulation by the sodium ions and sufficient depolarization of the membrane has taken place, it means more sodium ions have actually entered. The receptor potential becomes large enough, which can stimulate the voltage-gated calcium ion channel proteins to open, right? So we need the influx of sodium ions to open these voltage-gated calcium ions, right? So these voltage-gated calcium ions would open. As a result, what would happen? The calcium ions would enter the cytoplasm of the chemoreceptor cell, and they would stimulate the exocytosis of vesicles, which contain a neurotransmitter, right? And this would happen from the basal membrane of the chemoreceptor. So we'll just have a look at the picture in a while, but this is a sequence of events. Sodium ions diffuses in. As a lot of it diffuses in, this causes the voltage-gated calcium channels to open, right? And as calcium ions enter the cytoplasm of the chemoreceptor cell, it stimulates the exocytosis of vesicles, which contain these neurotransmitters, right? These neurotransmitters then stimulate an action potential in the sensory neuron. And the sensory neuron then transmits an impulse to the brain. So see, this is a sequence of events. So this is sodium chloride, which is in the food, right? It diffuses in, this is a microvilli. So it actually, if the sodium ions, they diffuse in, what they do, they depolarize the membrane in the sensory zone. So as they become depolarized, this stimulates the calcium ion channels which are here on the outside here. So they open, and this allows the calcium to enter into the cytoplasm here. What happens because of that? These vesicles, which are already there in the cell, right? They contain neurotransmitters. But what happens because of the entry of calcium, it causes these vesicles to fuse with the membrane. And through exocytosis, they release the neurotransmitters here. So this space that you see here is a synaptic cleft. So they release the neurotransmitters here, which get attached to this receptor here. Okay, so if the stimulation is above the threshold, impulses then travel to the brain along the sensory nerve. But if these impulses are not high enough, the threshold is not high enough, no impulse would be carried away, right? So remember, if the stimulus is very weak or below a certain threshold, the receptor cells won't be sufficiently depolarized. Sensory neuron will not be activated to send the impulses. But if it is high enough, it is strong enough, it would increase the receptor potential above the threshold value, right? And this will stimulate the sensory neurons to send the impulses. So this is actually what we call as all or none law, all or none law, right? So that's the threshold, it has to be above the threshold, okay? So only then the stimulus will be, the response impulse will be sent. If it is below the threshold, nothing would happen, right? So impulse is only transmitted if the initial stimulus is sufficient to increase the membrane potential above a threshold potential, right? So these threshold levels, they're not constant at all, right? They actually, for these to happen, and they often increase with continued stimulation. So you need a very great stimulus before any impulse is sent along the sensory neurons, right? All right, then now we are going to talk about the transmission of nerve impulses. As we were just discussing about depolarization, we will actually discuss about what an action potential actually is. So these neurons, they transmit electrical impulses, which travel extremely quickly along the neuron cell surface membrane from one end of the neuron to the other, right? Now, unlike these electric current, we just discussed, these impulses are not a flow of electrons, right? So actually what happens, this is actually the distribution of electrical charges change. Positive ions get in, negative charges go out, right? So these actually impulses, which we call as action potentials. So they occur via very brief changes in the distribution of electrical charge across the cell surface membrane, right? So these action potentials are caused by rapid movement of sodium ions and potassium ions across the membrane of the axon. So if you can recall the picture that you were just looking at earlier, right? So this is the rapid movement of these ions, sodium ions and potassium ions across the membrane of the axon. So in the same picture, so this is what happens in an action potential. See, the distribution of charges actually changes. So now let's talk about a resting potential. So we call a resting axon as one which is not transmitting any impulses, right? So when no impulse is actually being transmitted, we call that as a resting axon, right? So the inside of the axon is always slightly negative. So it has a negative electrical potential, right? As compared to the outside axons. So usually what happens, like there are positive charges, more positive charges outside as compared to the internal environment. So because of this relative charges, right? So the inside of the cell membrane is actually negative as compared to the outside. It is not that we have all the negative ions here. There are positive ions here. There is potassium, right? There is sodium, but there is more positive outside. More positive charges are outside here, right? And this is actually kept in this way. So this potential difference is usually about minus 70 millivolts, right? So it means inside of the axon has an electrical potential of about 70 millivolts lower than the outside. So this is why it's relative to the outside, it is comparatively negative. So this is what is known as a potential difference, right? And this difference is what we call as resting potential. It is maintained at this value, right? And then there are several factors which actually contribute to maintaining this resting potential. So if you look at this key here first, the yellow dots that you see, these are all sodium. And if you compare, this is the outside of the cell. This is the inside part of the cell. So if you compare these two, we have a lot of sodium ions outside, the ones in yellow that you see, right? And these kite-shaped structures that you're seeing here, the diamond-shaped structures, these are potassium. And if you compare potassium as compared to sodium, we have more potassium inside as compared to outside. So this is how this balance is maintained. Due to this, because actually this cell membrane becomes polarized. So we have more positive charges here because of more sodium, right? And we have lesser positive charges here. So comparatively, outside of the cell is more positive, right? And the inside of the cell is negative as compared to the outside, right? And here you are seeing a number of proton pumps here also. So this is a sodium potassium pump, right? This is a sodium channel. And you might also be noticing there are more potassium channels as compared to the sodium channel. So this is how the cell membrane is like. There are more potassium channels as compared to sodium channels. We have sodium potassium pumps. And this is actually what is actually working hard to maintain this resting potential to minus 70 millivolt. This is actually what is happening. So what this sodium potassium pump does, it sends 3 sodium ions outside, and at the same time, it brings 2 potassium ions inside, right? So actually, there are more positive charges going out as compared to what is coming in. So, and for this, actually, ATP is needed. So this resting potential actually is because of this membrane, which is selectively permeable. So it is more permeable to potassium ions as compared to sodium. So these sodium and potassium channels that you're seeing, they remain open all the time. Okay? Since there are more potassium channels actually there, so more potassium actually diffuses out, right? And like less sodium can actually come in, but more sodium can actually, more potassium can diffuse out. So these are the factors that actually contribute in maintaining that resting potential of minus 70 millivolt, right? So first and foremost are the sodium potassium pumps in the axon membrane, right? So these sodium potassium pumps, what they do, they pump sodium out of the axon and potassium ions into the axon. So as we just saw in this part here. So they would pump 3 sodium ions out. At the same time, they would take in 2 potassium ions inside. Now, since it's a sodium potassium pump, this pump, it uses energy from hydrolysis of ATP, right? Because it continuously has to move these ions against their concentration gradient. And as we say, against their concentration gradient, so you can see there are lesser number of sodium ions here, right? And they are actually being pumped out against the concentration gradient, right? And here, if you see here, so we have less number of potassium ions here, but they are also pumped inside against the concentration gradient, right? So these pumps, they require ATP to work. Besides the sodium potassium pumps, there are many large negatively charged molecules inside the axons as well. So because of these anions, what happens, they attract the potassium ions because there are more potassium ions inside. So they attract them and they reduce their chance of diffusing out of the axon. So as we were discussing, if there are more potassium channels and potassium can easily diffuse out. So the diffusion is also stopped because of they being actually making bonds with anions. So through this procedure also, the diffusion is actually stopped. Then this membrane is impermeable to certain ions. So mostly what happens, sodium ions, they cannot diffuse through the axon membrane when the neuron is at rest. So here also if you see, sodium is actually being taken out only through this sodium potassium pump, right? But it cannot diffuse out on its own. Potassium, you see, it can diffuse out, right? But sodium cannot diffuse out, right? So this, once a sodium ion is inside, it cannot go out. So this membrane is permeable, right? It doesn't allow sodium to go out, but it allows potassium to go out. So selectively permeable. And besides these channels for sodium and potassium, there are voltage gated channels as well, right? So these voltage gated channels remain closed, right? And actually, like these channels are closed. So what happens because of this, sodium and potassium ions are also actually, this keeps them from diffusing through the axon membrane. So these voltage gated channels, the moment they open is the time when an action potential is generated, right? So when we talk about an axon at rest, right? So sodium potassium pump would be working hard to take the sodium out and bringing the potassium in. These large negative anions would make bonds with potassium ions and would stop them from diffusing out, right? This axon, right, it would not allow the sodium ions to go out once they're inside, right? And these voltage gated channels would remain closed, right? And this would also stop the sodium potassium ions to diffuse through the axon membrane. So these are the factors which contribute towards this resting potential, which remains at minus 70 millivolt. So as we were discussing, this membrane has protein channels for potassium and sodium, which are open all the time. So we have different type of protein channels here, right? So there are some which remain open all the time, and they are more for potassium than for sodium, as we just saw in this diagram here. So they're more for potassium as compared to sodium. So therefore, because of that, what happens, some potassium diffuses back out again much faster. As it comes in, it diffuses back again, right? Faster than sodium diffuses back in. In addition, there are many large negative charge molecules that we discussed that would attract these potassium ions, right? And they would reduce their chance of diffusing out. So because of all these effects, the overall excess of negative ions inside the membrane is actually what makes this inner environment more negative as compared to the outside. And this membrane is relatively impermeable to sodium ions, right? But what happens since there is more sodium outside already? So this actually creates a steep concentration gradient, right? So there's a lot of sodium ion outside, and you know that it can actually flow from a high concentration gradient to a low concentration gradient, right? So this develops a very steep concentration gradient, and we know these protein channels, they remain open all the time, right? And the second part is that the inside of the membrane has become relatively negatively charged as compared to outside. So this also attracts the positively charged sodium ions, right? So because of these two factors, sodium would diffuse in, and this is what we call as a double gradient, right? So there is a steep concentration gradient. We have a lot of sodium outside, right? And then because it's negative inside as compared to outside, the positive charges will be attracted. So this actually what creates an electrochemical gradient, right? So it becomes a double gradient. So see here, we have this sodium channel, but you see there is a lot of sodium ion outside. So it can easily come in because of this steep gradient from high concentration to lower concentration here, right? And then this part here is comparatively more negative. Inside of the cell is more negative as compared to outside. So this creates an electrical gradient also, right? So this is what we call as electrochemical gradient.